Okay, the next part of looking at the immune system we're going to look at is referred to as cell-mediated immunity. So this would be the sort of second pillar of the um, immune response. We've looked at AMI, so now we're going to look at cell-mediated immunity referred to as CMI. Now there's the take-home message here, four basic things that you're going to learn. We're going to learn about. First, immunity can be conferred by transfer of cells. What we've learned up till now is that immunity is transferred by the transfer of antibodies. If I transferred antibodies from one person to another, I can transfer their immunity, AMI immunity, at least temporarily to that other person. And if you did that through cells, you could do the same thing. Two antigen, in this case, you'll see you can do it through cells as well. Two antigen-specific and non-specific cells are used, and basically some of the cells we've already come across. Three, CMI is very important in clearing intracellular pathogens, and it's also important, I should have written on here, in clearing malignancies as well. And fourth, really exciting development in the last couple of 2 to 3 years are that the recognition that T cells can be modified and used as personalized anti-cancer for personalized anti-cancer immunotherapy. So T cells using the ability to go after can be to go naturally go after malignancies can be engineered to and we'll see that later can be engineered to provide a new therapeutic route that's very exciting. So if we look at immunological responsiveness, AMI takes care of extracellular pathogens, bacteria or bacterial parts. These are things that are outside. But they are not the only thing that can infect you. The things that can infect you are things like viruses, for example, or bacteria that can reside, like mycobacterium, that can reside in inside cells, or even cancer. And so CMI is all about intracellular pathogens, virus-infected cells, tumor cells, and also, as we will see, foreign grafts during transplantation. Generally, AMI is mediated by soluble antibodies, and CMI is mediated by T lymphocytes. T lymphocytes here are the big players. So the two types of CMI-based lymphocytes, there's two types. Uh, One, effector cells with direct cytotoxic activity. They can wipe out another cell if they've located it. And effector cells that are involved in a type of hypersensitivity reaction that are involved in those referred to as delayed hypersensitivity type 4. We'll talk about those later. The most important is that there's a direct cytotoxic activity. So this direct cytotoxic activity that refers to this direct cytotoxic activity refers to means that direct lysis of the cells is carried out. This involves antigen specific T lymphocytes, and we've seen how that can happen through the T cell receptor. And antigen non-specific cells such as macrophages and NK cells. And I want to point out here that macrophages are antigen non-specific cells. The only antigen specific cells are T lymphocytes and B cells. Now this response is directed to chemically conjugated cells. This can happen as you'll get a T4 hypersensitivity reaction, as we'll see later. Virus infected cells, malignancies, and graft rejection. Now we can look at these cells. The most important are the T cells, and as we've seen before, they're characterized by CD molecules on their surface. And one of those molecules is CD3, as we've seen. We've seen CD3. CD3 in turn, it turns out, is on all mature T cells. It's on all mature T cells. CD4, as we've seen, is referred to as T helpers. These are T helper cells. Okay, and we've already seen how they play a role. CD8 is the new one, the new kid on the block. These are referred to as T killer cells or cytotoxic T lymphocytes. Um, Cytotoxic T lymphocytes, T killers. So these molecules, this is the new T cell that we're going to talk about. You'll see become important. So T helper cells recognize antigen, which is presented in association with class MHC2 molecules, thus directing antibody production, and we've seen how that happens, plasmacytes and memory. T killer cells recognize antigen when they're presented not by MHC2, but when they're presented by MHC1 molecules. Okay, so this is MHC2. T killers, CD8 positive, interact with MHC1s, and they direct the cytotoxic response. And again, they're referred to as cytotoxic T lymphocytes or CTLs. So these are MHC1 and these are MHC2. T helpers interact with MHC2. T killer cells, CD8 positive cells interact with MHC1. An optimum CMI response requires antigen presentation by phagocytes by the following types: monocytes or macrophages. Monocytes are basically macrophages, phagocytic cells that reside in the blood. Macrophages in the tissues. Dendritic cells. Dendritic cells reside in the secondary lymph, the primary, sorry, um, secondary um, tissues of the immune systems, like lymph nodes and thymus. Langerhans cells, which reside in the skin. Marginal zone macrophages, which reside in the lymph nodes and the spleen. Follicular dendritic cells, which also involve in these high areas of high B and T cell concentration. One other thing, and antigen processing cells process antigens by presumably proteolytic processes, and they also synthesize IL-1 or interleukin-1. So that's those are the characteristics that you see from these antigen processing cells. The key thing here is that all nucleated cells express MHC1. That means liver cells can express MHC1, brain cells can express MHC1, muscle cells can express MHC1. All nucleated cells can express MHC1. There's only one cell type that doesn't, and those are non-nucleated cells, and those would be erythrocytes. And so everything else does. So all B cells, T cells, muscle cells, brain cells, liver cells, heart cells all express MHC1. If it has a nucleus, it does. That's not true for MHC2. MHC2 is expressed by only antigen processing cells and there's a few other types of cells, but it turns out that's not involved. They're not involved in the immune response really too much. So let's look at there are two phases of this response. The first phase is referred to as the sensitization phase, and this is where an antigen processing cell, and in this case, it's going to be generally is a dendritic cell, for example. One of these can be a macrophage, but generally it's a dendritic cell, and express B7 and MHC2 peptide. So it's gone through, it's taken the peptide antigen in, it's processed it, it's put it on the surface, and that's going to interact, as we've seen, with CD4, a Th1 cell. And a Th1 cell, as you see, was generated where it was generated in the response that we saw before, where you get Th1 and Th2 cells. And what that's going to lead to is the production of IL-2, which we've seen before, and IL-2 receptor on the surface of the Th1. They actually produce the interleukin, produce it, then consume it with the receptor. The receptors are actually internalized, and that further activates those cells. But it also produces IL-2 that can activate and interact with other T cells. Because fundamentally, if you can't produce IL-2 or IL-2 receptor, you can't, particularly IL-2 receptor, um, um, you can't respond to IL-2. So you get this consumption and then further proliferation of these Th1 cells and clonal expansion differentiating into Th1 cells. So these guys are getting cloned out and further expansion and proliferation has occurred. Now, uh, when this happens, uh, you by and large, and this can happen on the same cell, but in the vicinity, there will be virally infected cells or abnormal target tissue, and there'll be cells that are referred to, they'll wander around, but they're referred to as pre-TK cells, pre-T killer cells. They express IL-2 receptor on their surface. There is MHC, the peptide that's being expressed on the surface is being expressed now, not in the virally infected tissue. Could be brain tissue, could be liver tissue, could be a dendritic cell, could be anything, because all nucleated cells can produce IL-1. And so therefore the MHC1, all nucleated cells can produce MHC1, not IL-1, sorry. This MHC1 peptide is then presented, interacts with the T cell receptor, and this MHC1 interacts with CD8 and not CD4. And so therefore you get interaction of a pre-T killer cell sensing and probing and finding out, oh, here's the MHC1 peptide on this virally and this on this tissue. Now the IL-2, sorry about that. The IL-2 that's produced by that Th1 cell activation will go to support and actually activate these pre-T killer cells. And so the CD4 is important because what it does is it provides this interleukin IL-2 to go and bind to the receptors on the surface of the pre-T killer cells and activate those cells. Now, what happens during this antigen presentation process? This process is a bit different than for MHC2. Because for the MHC2, what you're doing is you're dragging in an antigen from the outside. It has to be processed. You have to have endosomes. You have to process that. You have to go through exchange, then the peptide or the emerging with these lysosomes. But for virally infected cells, remember the transcription and translation of those proteins is happening inside the cell. Okay, there's nothing outside the cell that's necessarily happening. The cell that's in there is going to have essentially the antigen is inside the cell already. And so in this process, lots of times there'll be a good chunk of those viral proteins will be produced, and there's a very large complex, a proteolytic complex called the proteasome, and this complex it's sort of it's in some respects it's sort of like the garbage disposal of the cell. Proteins that become damaged, proteins that are potentially tagged, you don't want hanging around very for a long time, will be cut up into peptides and be catabolized by the cell. So this is a way that the cell kind of cleans up itself, cleans up the proteins in itself that are damaged, etc., have gotten signals and to be removed. And it produces a number of peptides that from that particular from a not a lot of proteins in the cell, but also from that from the internal antigen. That could be the capsid, for example, of a virus. Now, what ends up happening is, in this case, this happens in the endoplasmic reticulum, the rough, and there is a molecule called calreticulum here, this green, and MHC1 is actually one peptide, and it has beta microglobulin that's attached to it. We'll look at the structure in a little bit more detail. And then it also is connected with a protein that supports and stabilizes the MHC1 pep protein. And there is also then a transporter that will sample in which the peptides can come into here. Now, when these peptides from the internal antigen come in, they're sampled by the MHC1s that are produced by those by the cells. And if one of those MHC1s, and there are actually several MHC1s that are produced by cells, uh, is able to bind to a peptide fairly tightly, it's going to be transported to the plasma membrane via the Golgi to get, and it will be taken out. The tapsin will be released and removed, as well as the calreticulum. And that peptide MHC1 complex is going to move then to merge to the surface and display then the MHC1 on that surface with its peptide. Okay, so proteasome peptides flow into the rough endoplasmic reticulum. They bind. This leads to transport to, uh, to uh, um, endosomes that are going to allow it to be then transported to the surface so it can be sampled. And so if a cell is infected, it will display peptides from that antigen on that surface. So let's stop here. We'll stop this segment. You can take a look at this. Stop the um, tape, and then the answers will be on the next slide. Okay, so now let's look at the MHC1. So a little bit more detail. So MHC1, as I said before, has is actually one polypeptide. Unlike MHC2, it's composed of two. You have one chain called the alpha chain, and then beta microglobulin, which is a structural protein which will bind to the MHC1. And you can sort of see this little cartoon here. Here's the MHC1. There again is a pocket for a peptide, and the beta microglobulin are there to structurally support the protein structure. Now, if we look down into an MHC1, what we'd find is that the binding pocket is composed entirely of that alpha chain. And again, it has we've seen this before, it has beta sheet structure across it to form the bottom. And then on the top part, it has um, alpha helical chains that serve as almost like teeth biting down on that peptide. We can sort of see the differences here. So here's MHC1, in which you can see the blue is all one chain. And if we look at MHC2, what we find is that when we're looking at MHC2, now you've got two polypeptides, in which the binding site is composed of the two polypeptide chains, as you can sort of see here. So only that chain, only the MHC1 they can bind to that peptide is selected and moved to the surface, and it does it again by the beta sheet floor amino acids interactions as well as this alpha helix. Sort of gives you appreciation of that. If we were to look down, if we look down at them, what we find is that they look very similar. You have the alpha helix on both, the peptide, and you have a beta sheet floor. But you do notice one thing: you can generally the MHC1s bind shorter peptides than the MHC2s. They're capable of binding bit longer peptides, whereas MHC1 binds shorter peptides. Now, if we look again at the complex with the T cell receptors, a crystal structure, we can see again it's the similar thing. The floor, there's binding interactions with the floor. I'm going to stop it here with the floor beta sheet floor, interactions with the side of the peptide from the alpha helical teeth. And then the T cell receptors you can see here is sampling, it's touching the solvent accessible part of the peptide. And then there are other linker loops, as you can see here between these two chains. There are other loops that bind to the like to bind to this particular MHC1, and so that's another thing that's critical. The T cell receptor here, you've got the two chains, but the T cell receptor only will recognizes the peptide as well as the MHC1. And so that's what's selected for. Again, the T cell receptor for the CD8 cells, similar to CD4 cells, is one T cell receptor, one type of ability to bind, one type of peptide MHC, and there can be some variants, but MHC1 peptide it's interested in binding. And in the loop regions, the loops really bind to solvent accessible surface for the peptide. Now, when the pre-T killer cell actually interacts with IL-2, it now forms a memory cell. It will divide and form one of those will be a memory TK cell. So this allows you to have memory of what's going to happen. And then it forms what's referred to as the a fully activated T cell, referred to as the C cytotoxic T lymphocyte. Always like, this probably dates me, always like to think of these as um, the terminators, terminator cells, because these guys can kill things. They can go out, seek, and destroy things. And so this IL-2 really activates and further activates these these types of CD8 cells, these CTLs. And what is noticed to them is that you'll start seeing granules, what are referred to as lytic granules, inside the cell. These are going to be filled with noxious things, enzymes, particularly enzymes that are going to be dumped in pore forming molecules, dumped on the surface to kill the cell. It also contains this molecule like fast ligand, which is a death signal. It sends a death signal to the cell to die. So that, just to recap, all those events from the CD4 activation to CD8 pre-T killer cell activation to a CTL composition are referred to as the sensitization phase of CMI. They're sensitized. The effector phase, essentially the killing phase of this, is that when the T killer cell then recognizes and binds to a virally infected cell or a normal cell, it's going to initiate programmed cell death by one way, as these granules, which are filled with really noxious things, will move to the surface between the two molecules. These will be dumped into the surface, into the targeted cell. and this is referred to as a cell membrane rearrangement occurs, and the granules are moved to that surface for their job. Another part is the Fas ligand will bind to Fas, and these death receptors, what they will do is it potentially could cause the cell death of the virally infected cell too, by initiating apoptosis, which is basically the um, destruction button of the cell, in which there's like T minus 9 seconds and counting before you're destroyed. Now these granules that are dumped on the surface contain esterases, proteoglycans, and the molecule we're going to see a little bit later, perforins. Now, when this happens, these perforins, lytic enzymes, do they work? The perforins form um, channels within the membrane. The lytic enzymes go about destroying proteins, and as a consequence, what happens is the T cell will move away from the cell it's dumped this stuff onto. That cell will lyse further and be destroyed. And then the T killer cell is going to go out and seek out and try to destroy another cell. And they can do this multiple times before they kind of wear out and die. So let's stop here. Here's a set of review questions. We will stop here, and I think I'm going to stop here. The questions the answers are on the next slide, so you can answer the question. I want to do that now. And then the answers are on the next part.